Alzheimer's disease and other neurological diseases that have amyloid protein as one of their signatures are really difficult to study. Being able to actually see these structures and how they form is just impossible for standard imaging. You can't see these things naturally if you just put them under a microscope, so you have to tag them. You have to label them with a special type of molecule that'll glow. All of these little tags or all these little labels are glowing all the time, and the resolution of the microscope just kills you and you can't see the actual structure of what's going on. What if we actually used a sensor, a little molecular marker that we know can detect amyloid proteins, but not permanently attach it? And what's amazing about these sensors is that they're dark when they're just in water, but when they randomly collide with the protein that we're interested in looking at, then they glow. You can think of little fireflies that only turn on when they're right next to the amyloid protein that they're targeted to. One of the things that we can really see very well with our technology now are these long fibers that are the precursors of the big plaques that you might see from patient samples. Maybe we can actually design therapeutics that actually can attack the disease at a more basic level before too many cells are killed.